And welcome to the Research Works podcast, brought to you in association with Curtin University and the Healthy Strides Foundation. Your hosts are Dr. Dana Poole and Dr. Ashley Thornton, and together we will interview world-leading researchers in the area of child health to support your practice in being more evidence-based.
Welcome to another episode of the Research Works podcast, everyone. We have another wonderful article and guest to introduce to you all today. On the show, we are very lucky to be joined by Dr. Michael Trevarrow, who is joining us all the way from Nebraska in the United States of America. Welcome, Michael.
Welcome.
Thank you guys for having me.
Well, it's so cool. I think it's so amazing that we actually do have the technology that enables us to speak in this kind of way. I think it's the greatest thing about Zoom and all the new technology we've had to live with the COVID world.
Absolutely.
And so today we'll be talking about a really interesting paper that I know you guys are all going to love. It was recently published in Developmental Medicine and Child Neurology in just March 2021, and it's titled Microstructural Changes in the Spinal Cord of Adults with Cerebral Palsy. And here Michael was the lead author of the paper.
So just a little bit of an introduction for our listeners. Dr. Michael Trevarrow is currently a postdoctoral research fellow at the Institute for Human Neuroscience at the Boys Town National Research Hospital in Nebraska, USA. His research focuses on utilising neuroimaging and non-invasive brain stimulation to study the underlying neurological abnormalities of the brain and spinal cord that contribute to impairments within the sensory motor system. His expertise in neuroscience and motor learning and control are certainly of great interest to us as clinicians.
Oh, I think we can learn so much from neuroimaging. And as clinicians, we often see the functional output all of the time in our work. And neuroimaging can provide such a rich source of information to help us, really understand what we're seeing and potentially what we can be influencing in our therapies. Because ultimately, when we're talking about neuroplasticity, that's what we're talking about. So before we go into all of that, though, Ash, what have you got for us today with your icebreaker?
So this icebreaker question, when I was trying to decide which one, I picked this one and then immediately had several answers to it. So I'm keen to hear what you and Michael have as answers to this. Our icebreaker question for today is, what are you notoriously bad at?
Oh, Michael, tell us.
That is a great question. I actually thought of something right away. I'm notoriously bad at keeping track of things. So my keys and my debit cards and wallet, I have lost way too many times.
Oh my goodness, that sounds.
That is what I am notoriously bad at. Pretty much my whole life.
At least you're consistent, though.
Yeah.
It has definitely stayed consistent.
What about you, Dana?
Oh, me? Okay, well, that's going to be first. I am really bad at lying. I had this, when I was younger, I was about 17 and I had my first mobile phone and I had to call the phone company on behalf of my mum because it was under my mum's name.
Right.
So when I called up, I just couldn't lie. And I had the full intention to pretend that I was her, but I forgot her full name. I forgot her birthday. I got it wrong 3 times. And then the lady on the phone is like, "You are not who you say you are you?" I'm like, "No, I'm not." So I'm terrible. I just don't get my facts straight if I'm trying to lie. So that's
kind of reassuring.
Yeah. I can't lie to your face because you can just tell on my face.
So you'd be really bad at poker as well,
it'd be so terrible. I'm like, I've got a secret.
The big grin on your face.
Yeah, that's right. So yeah, that's my worst part of it. Yeah. What about you, Ash?
Again,
curious.
This was a very easy one for me. Directions and sense of direction. I am notoriously bad at knowing where I am, knowing where other things are around me, especially when I go somewhere new. Like my husband has a really good sense of once we're in a place, he'll know exactly where everything is from that place. And it really infuriates me because I'll walk out of a hotel and then turn left and walk confidently in that direction. And where we need to go is completely the other way.
That's exactly how I am in my city. I'm pretty good at directions, but then once I get outside to a new place, it all falls apart.
Yeah. So thank goodness for the little navigation app on my phone.
Yeah. So if I got you right now to point to where's north?
Oh, no. I have no idea.
I don't know where I am.
Especially because our room has no windows. So I'm getting better at that kind of thing. But yeah, no, directions is my kryptonite.
I feel like that was a great question. We've learned a lot about each other there. Weaknesses, bang.
Yeah. Okay, so once again, I feel like we've kind of learned a lot about everyone. So now that we've done that, Dana, why don't you introduce the paper to us?
I will indeed. And there's a lot of really big words in this one, so just bear with me. So to quantify the microstructural differences in the cervical-thoracic spinal cord of adults with cerebral palsy, 13 adults with a mean age of 31 years and 11 months and 16 adult controls without cerebral palsy with a mean age of 31 years and 4 months underwent magnetic resonance imaging or MRI of the proximal spinal cord, so from C6 to T3. The variables of interest include the cross-sectional area of the spinal cord, the grey and the white matter magnetization transfer ratio, the fractional anisotropy of the cuneatus, and the corticospinal tracts were calculated. The results indicate that the total spinal cord cross-sectional area and the proportion of the spinal cord grey matter cross-sectional area were significantly decreased in adults with cerebral palsy. Functionally, participants who had worse hand dexterity clinical scores as measured by the box and blocks test had reduced grey matter. So what this means is that the microstructural changes do play a role in the extent of the hand sensory motor deficits that are seen in adults with cerebral palsy.
Thank you, Dana, and well done for getting through.
I was sweating a bit.
Michael, I don't know how you go doing presentations on all of this work and getting through all those big words.
Definitely have to make sure I know how to pronounce them.
Yeah, hats off. So this is actually a really significant study because as we understand it, there hasn't really been any studies investigating those microstructural changes that we're talking about within the spinal cord in people with cerebral palsy. So we thought we'd kind of go back a step and just start off by getting a bit of a summary about current knowledge. So, you know, what we know about functional decline in adults with cerebral palsy and go from there.
Yeah. So cerebral palsy is often defined as a non-progressive disorder. Yeah. And really what that is referring to is the neurological insult to the developing brain. Mm-hmm. And so because of a number of factors such as weight gain and increases in fatigue and accelerated sarcopenia or the weakening of muscles, all of these factors kind of contribute to a progressive deterioration of the musculoskeletal system within this population. Mm. And so many individuals with cerebral palsy, as they transition into adulthood, actually may lose the ability to walk independently or may require an assistive device to walk when they didn't before. Mm-hmm. And so these deteriorations in the musculoskeletal system can then inherently impact the upper extremities as well. Mm. And so this can lead to difficulties and challenges in completing tasks of daily living. Mm. And that is inherently going to increase stress in daily life and can contribute to a decrease in the overall quality of life. Mm. And so really studying this adult population in individuals with cerebral palsy is really critical. A lot of research has really been focused on the pediatric population.
That's right.
But the transition into adulthood is very important.
Absolutely. And, you know, in this paper, you touched on some of the previous imaging work that demonstrates that there are, you know, those activity-dependent neuroplastic changes that often occur following the brain injury. And this was quite significant in terms of, you know, what we know about neuroplasticity, wasn't it?
Yes. Yeah, definitely. And so some really interesting work has come out by Dr. Kathleen Friel. Mm-hmm. She showed that in an animal model, in response to inactivating one side of the brain, the ipsilateral corticospinal tract connections can actually be maintained. Mm. And then so we also see this in individuals with unilateral cerebral palsy, in which when there's an insult to one side of the brain, then those corticospinal tracts that are controlling the more affected side of the body are actually shifted toward the contralesional hemisphere, which this type of reorganization is not really seen in the somatosensory system. And so what this means is that the corticospinal tracts that are in the side of the brain that is controlling the affected side of the body is different from the side of the brain that is processing the sensations from that side of the body. And so this can really lead to a dissociation in sensory motor processing and difficulties in sensory motor integration. And individuals that show more of this reorganization actually tend to have increased sensory motor impairments. And this kind of highlights the idea that these changes that are occurring within the brain don't just affect the brain, but really they're having downstream effects at the level of the spinal cord and out to the peripheral nervous system.
And I think that's just so interesting when you talk about that work. I mean, as soon as you say, as soon as you talk about that, I think we all sort of relate to it really quickly about our treatments that we already provide. We talk about constraint-induced movement therapy and, why it is that we do what we do. It's come from this kind of work and it's this understanding of reorganization of the corticospinal tract. So that was a great summary. Thank you so much. I'm going to listen to that a few times, I think, to get that in there. So now just moving along to why, in terms of the spinal cord, why are the microstructural changes of such interest in people with cerebral palsy? I guess this kind of sparks why you did this, this paper.
Yeah, definitely. I think the spinal cord in general is of particular interest because there are a lot of functional and structural neuroimaging studies that have shown alterations in abnormal structure and function within the brain and kind of connected these with different sensory motor processing deficits within this population. But one thing that they're kind of lacking is, well, the sensory information and motor processing commands are inherently going to be going through the spinal cord. And so without knowledge of what is actually happening at the level of the spinal cord, it makes many of those studies kind of difficult to interpret, or at least not really giving a comprehensive picture of the disorder. And so that kind of sparked interest in trying to identify what's actually occurring at the spinal cord and how is this affecting some of the impairments that are seen within this population. Yeah,
sure. So no, really sort of bigger sort of, high level kind of thinking then. So can you just help us to differentiate between, what does the gray matter represent? And then from there, what does the white matter represent in the spinal cord?
Yeah, so the gray matter in the spinal cord, or in the brain for that matter, is going to be representative of the neuronal cell bodies. And so the number of cells, as well as the size of the cells, and the dendritic arborization, which is kind of a term for how many synaptic connections there are, these things are all going to impact the amount of gray matter that is seen within the spinal cord. And then white matter, on the other hand, is really reflective of the myelination around the axons of the neurons. And so that's important for sending signals along neurons. And many other movement disorders have shown deterioration or degeneration in the myelination of neurons. So that's why white matter is also of interest to us.
Yeah, that's a wonderful summary. So now moving on to sort of, I guess, the outcome measures that you used in your study. Obviously, imaging is really not our area of expertise.
Definitely not.
So I thought if you could just help us talk through why, first of all, on a really high level, why did you use MRI, and what does MRI enable clinicians and researchers to see?
Yeah, so structural MRI is a really valuable tool in identifying abnormal or different structure within the brain, or in this case, in the spinal cord. And so at the gross level, this can mean identifying things like malformations, but we can also use MRI to segment tissue, meaning kind of understand the amount of gray matter and white matter and cerebrospinal fluid, and to identify how this is different within the brain and spinal cord is going to really give insight into how these things are impacting functional deficits. And so MRI has really a lot of utility in that area.
Yeah, and I think you do get so much information from that. So one of the outcome measures that you sort of were looking at was gray matter and white matter cross-sectional area. So how do you do that?
Yep. So cross-sectional area is just referring to essentially taking, if we were to take a horizontal or an axial slice of the spinal cord.
Yeah.
And then we used the spinal cord toolbox, which is a great semi-automated tool that helped us to process all this data. And using that, we could segment out the gray matter and the white matter within each of the slices of the upper spinal cord. And so from there, then you can quantify how much of that slice or of that cross-sectional area is actually the white matter and the gray matter.
Sure. This, I'm just going to just digress a little bit here. This reminds me of when I did my PhD and I was looking at muscle volume, and it was we used MRI as well, and it was cross-sectional area. And I had to calculate overall volume. So you segment it and you add it all up together. So I can only imagine how many hours upon hours of work goes into getting that data.
Yeah.
It's a fair bit of work there, Michael.
Yeah, there's a lot of work that goes into it, but really utilizing the spinal cord toolbox makes it really simple. It's a lot of functions that can be run, and a lot of these processes are actually automated.
That's amazing.
So kind of a key before processing all the data is, of course, acquiring good data.
Yeah.
So
That's right.
ensuring that subjects are still situated within the MRI is going to be extremely important, because if that doesn't happen, then on the back end, there's not much that you can do with that data.
Yeah.
Yep. So that does also just show that the data that you did have was very, was accurate, because you were able to actually draw that data out, which I think is a really good point to make.
Absolutely.
When you're looking at the graphs. Okay, so the word that I sort of struggled with at the beginning, if you can just describe what this actually means, fractional anisotropy, what is that?
So fractional anisotropy is an outcome measure that we get from diffusion weighted imaging, or DWI. And so DWI is an extension of MRI that allows us to look at the integrity of the tracts within the brain and within the spinal cord. Right. And so fractional anisotropy is measured from 0 to 1, and 0 is reflective of complete isotropic movement of water molecules, and then a score of 1 would be complete anisotropic diffusion of water molecules, or diffusion all in one direction. And so generally speaking, the higher the number is, or the closer to 1, then the more intact those tracts within the nervous system seem to be. And so, for example, previous studies have shown that a loss of fractional anisotropy within the thalamocortical tracts in the brain in individuals with cerebral palsy is actually related to decreases in things like sensory motor functioning and mobility.
Wow.
So it's a really valuable tool to kind of study those tracks.
It is.
Yeah, it's really interesting. And the last one to go through is the magnetization transfer ratio, the MTR. What does that mean?
So that's yet another extension of MRI, magnetization transfer imaging. And from there, in general, the MTR, or the magnetization transfer ratio, gives us kind of a gestalt of potential damage to gray matter and to white matter. And it also has been shown to be related actually to inflammatory responses within the CNS. And so this is something that's been studied in other disorders such as multiple sclerosis, and then also in individuals with spinal cord injury. And so it kind of allows us to make more inferences about the structure of the spinal cord.
That's really cool.
Yeah. I feel like I've learned so much already.
So have I.
Michael, just to kind of describe your participants in a little bit more detail now. Your group of participants had max levels of between 1 and 3. They hadn't undergone any upper limb surgery or botulinum neurotoxin A injections in the year previous. So something we noticed that you did, which will be most familiar to our listeners, and it was a relief to read something that I actually understood in a bit more detail, was that you used the box and blocks test, which, as we know, is a measure of hand dexterity. Could you explain to us why you chose to use the box and blocks?
Yeah, sure. So for a variety of reasons, the box and blocks is, first of all, a pretty simple, straightforward task, and it's very quick and easy to do. But more importantly, it's a task of upper extremity and hand functioning. And so the reason why we want to look at upper extremity functioning clinically is because we're measuring the upper spinal cord within the MRI. And so the portion of the spinal cord that we're measuring, as we said, C6 to T3, is the portion that is going to innervate or control the upper extremities. And then so doing a clinical test of hand dexterity is then going to allow us to try to directly connect the functioning at that level with the structural changes within the spinal cord, the portion of the spinal cord that is controlling that area.
Yeah.
And I love that you did that. I think that is so cool that you did do that. So many times it's hard to draw relationships between what you find in neuroimaging to actually what it means. And by doing that, I think it just draws such great information that you can apply straight away. So like the clinical implications become very evident. So yeah, that's awesome that you did that. So now to talk about the results, we're really excited to go through these with you. So the first one, cross-sectional area, you want to go through that result with us? I'll get you to say it because I think it's really cool when you describe it.
Sure, yeah. So we found that the total cross-sectional area of the spinal cord, as well as the gray and the white matter cross-sectional area, were each significantly reduced or smaller in the adults with cerebral palsy. And so the total cross-sectional area being smaller is quite literally showing that the spinal cord is smaller in these adults with cerebral palsy. And then the differences in gray matter are suggesting that the total number of cells or synaptic connections within this area of the spinal cord that we're looking at is also decreased. And then I think an important thing to point out is that after we normalized these measures, the gray and the white matter, to the total cross-sectional area, we found that it was the gray matter cross-sectional area that was really kind of driving this effect. So that was still significantly different between the groups. And so I think that there are a number of possibilities as to why this is reduced. One possibility is that, previous studies have shown that in response to an insult to the brain, the corticospinal tract terminations can actually shift a little bit. And so if these are shifting to terminate more in the dorsal portion of the spinal cord, where the sensory neurons are typically residing, this could potentially lead to competition for space and for real estate within that section of the spinal cord and lead to an overall pruning or reduction of the cells. And on top of that, another possibility is that this reduction in cells is reflecting a decrease in the GABAergic inhibitory interneurons. So previous work has suggested that individuals with CP demonstrate increases in excitability within their spinal cord. And so if this reduction in gray matter is representative of a reduction in GABAergic cells, then this could make that connection make a little more sense.
Yeah, that's really interesting, actually, isn't it? When you think about the reasons why and
the implications.
Yeah.
So, Michael, the magnetization transfer ratio you found was also significantly lower in the corticospinal tracts of adults with cerebral palsy. What does that mean? Yeah,
so that can suggest a few different things. I think, first of all, it's showing that there might be some sort of damage to the actual gray matter and the white matter within the spinal cord. Another possibility, I mentioned that the MTR can be representative of inflammatory responses. And so in individuals with cerebral palsy, it's not completely uncommon to see chronic inflammatory responses. And so this can also lead to an overall reduction in the number of cells. And interestingly, the MTR was actually related to the amount of gray matter within the spinal cord. And so the MTR kind of could be reflecting these inflammatory responses, and then that relates to a decrease in the overall number of cells and the reduction in gray matter.
Yeah, and this is probably outside of the scope of your study, but is there any way that you know of to kind of identify those inflammatory responses that you're talking about?
So that is, a little outside of my scope, but I think that's definitely interesting work to kind of identify whether these chronic inflammatory responses are contributing to not only changes at the level of the spinal cord, but also some of the sensory motor impairments that are seen within this population.
Yeah, it's really interesting.
And I think with inflammatory response, I think in my head when you say that, you always think it's more of an acute thing that you might see, but you really are referring to something that's more chronically inflamed, aren't you?
Yes, yep. And that can kind of lead to long-term changes within the nervous system.
Yeah. And then finally, the correlation between the box and blocks test and the grey matter cross-sectional area, as well as the total cross-sectional area. Could you tell us a little bit about what you found there?
So I think that this was one of the more interesting parts of the study. As we talked about before, identifying direct relationships between the alterations in the microstructure of the spinal cord and actual clinical outcomes is really showing that these microstructural changes actually might be contributing to the sensory motor impairments that are seen within this population. That being said, I think that a lot of future work could seek to identify whether there are other clinical tests that are also being affected by these changes that we see within the spinal cord.
And the box and blocks test is so great though, because it does integrate, the sensory component of it, it's motor. It's such a great way to sort of understand what it is we might be seeing. So now I'm really getting to like the clinical implications of this now. we talk a whole lot about activity-dependent plasticity, and my background a lot is some of the work that you do in locomotor training, which is spinal cord injury rehab, and it is about providing the right kind of input so you're actually influencing at the spinal cord level, not necessarily at the level of the brain. Just speaking from a very gross level, right? Because you want to try to influence what's happening at the spinal cord. So, Michael, what do you think about the spinal cord in terms of the microstructural changes? Like, I think we sort of touched on it a little bit beforehand, but do you think that what we're sort of seeing as changes is something that's, maybe activity dependent use, what it is that actually the child or the person over time is able to contribute functionally and be part of and use a lot of activity that's contributing to those changes? Or do you think those changes really are more of a result of what happened to the original insult and, just we're looking at that sort of cortical spinal tract reorganization? Like, what are your thoughts on that?
I think that it's a little bit of both. I think that the direct changes that are occurring at the level of the brain are going to have downstream effects at the level of the spinal cord, kind of independent of environment and independent of what the developing child is doing on a day-to-day basis. But I also think that early intervention is going to be able to shape those different neuroplastic changes that we're seeing, not only at the brain, but also within the spinal cord. And it's also kind of a two-way street. And so we think about changes in the brain inducing changes at the level of the spinal cord, but it also could be the other way around, in that these kind of microstructural changes that are occurring at the level of the spinal cord could be impacted by therapeutic interventions and a child's day-to-day activities. And that could in turn potentially change some things that we're seeing at the level of the brain. So I think it's always kind of good to think about it from both perspectives.
Yeah. And then I think when you talk about your participants that you had, that the mean age was around sort of 30, 31. So there was, it's a good time point really, isn't it, to be looking at some of these changes and what they might mean?
Yes, definitely. Yeah. I think that assessing maybe how these changes are occurring developmentally and how the developmental trajectory of these changes from childhood throughout the transition into adulthood would definitely be of interest. Are these changes things that occurred at a very young age and haven't really shifted at all during the transition to adulthood, or are they actually changing alongside the functional changes and the deterioration of the musculoskeletal system that we see into the transition into adulthood?
And so I guess in terms of interventions and really thinking about, we're not just thinking about the brain, we are thinking about the spinal cord. And when you think about spinal cord, I think there are different kinds of interventions that you do need to think about that do influence and even things like, I love the use of electrical stimulation, what that might be doing, because we know it's providing sensory feedback up, and what that might mean in terms of trying to support any change that might happen from there. Just incorporate, I think being really specific about your intervention and knowing what structures you might possibly be influencing is a really important thing for therapists to be doing.
Yes, definitely. And I think that kind of understanding the changes that are occurring within the nervous system alongside the therapeutic interventions is really critical to kind of understand or get a better understanding of at what level are we actually making changes. And so if we're seeing improvements in functional abilities following a therapeutic intervention, are there actually changes that occurred within the nervous system that may be driving this? And I think that's definitely a very interesting topic.
Yeah. And even exploring like your max levels, because you had max levels of 1 through to 3. So these are individuals that are using their upper limbs as much as they possibly can, and as you describe it. So it'd be interesting to see what would be the case for, max 4 and 5, the role of repetition and practice. Things like that to consider. Is that something that you might look at?
Yes. Yeah, definitely. Definitely. I actually was considering that as I was kind of writing up the paper, and that it would be interesting to see if we had enough participants, of course, these changes that we're seeing kind of scale with severity of the disorder. So is somebody that has a max score of 1 going to present more significant changes in their gray matter as somebody that has a max score of 4 or 5? Yeah. And my guess would be yes, but that also requires a lot more subjects. So
Yeah,
Yeah, sure.
And I think you in the limitation section of your paper as well, you kind of highlighted that you're just focusing on one very specific subset of people with cerebral palsy, and there's obviously many other presentations to think about and look at with regards to sensory motor function as well. So I think there's lots of scope for different directions with what you're doing, and it's really exciting.
It is. It's really, really cool.
So, Michael, if I had to kind of get you to pin down one area for future research where you would like to see this work go, or maybe, where you're taking this work next, could you tell us a little bit about what that direction might be?
Yeah, so I think that this study kind of opens up a number of pathways for future work. A few of them we already talked about. So identifying whether these changes at the spinal cord are ubiquitous across different types and presentations of individuals with cerebral palsy. So is somebody that has hemiplegic cerebral palsy going to present different changes at the level of the spinal cord as somebody with spastic diplegic or a different type?
Yeah.
I think that's definitely an interesting line of work. Another thing is kind of, as I mentioned earlier, seeing what else the spinal cord has an effect on behaviorally and clinically. And so again, we only included one clinical test within this study, but including more clinical tests and seeing if these changes in the spinal cord are related to other things, that other impairments that are seen clinically will be very important. And then finally, importantly, I think the next step will be measuring and recording the lower spinal cord with MRI. And so the lower spinal cord is, of course, going to innervate and control the lower extremities and have a large role in mobility. And so I think that the next step will be evaluating the lower portion of the spinal cord in individuals with cerebral palsy to see if these microstructural changes are persisting.
Yeah. Lots of things to keep you busy.
Oh,
and
definitely.
And I think, often when we talk about and think about neuroplasticity, people, think about the brain. And so your work is really important in highlighting, not that it's so much bigger than that, and that the spinal cord actually plays a really important role in what we're talking about and those functional changes. So thank you for highlighting that, and thank you for talking to us about it today.
Yeah, huge congratulations.
Absolutely. Thank you.
It is very, very cool. All right, so we're going to go to our next segment, which is called Tell It to Ed. Now, as you all know, Ed is our producer who doesn't have a background in child health, but he listens in on all of these conversations and he's getting pretty good. But this topic, I can tell he's a little bit sweaty at the brow. So, Michael, I'm going to get you to summarize in about 60 seconds or so the work that you did here.
Perfect. Yeah. So what we did here was measure the structure of the spinal cord in healthy controls as well as individuals with cerebral palsy. And one thing that we showed is that recording MRI at the level of the spinal cord in individuals with cerebral palsy is viable. And we also demonstrated that there are specific changes in the structure of the spinal cord within this population. And so the spinal cord is overall smaller. And there are also differences in the amount of gray matter and white matter tissue within the spinal cord. We also demonstrated that these changes that we're seeing at the level of the spinal cord are directly related to some of the sensory motor impairments that we see within this population. And so future work should really focus on identifying what other roles the spinal cord plays in the different sensory motor as well as mobility deficits that are seen in individuals with cerebral palsy.
That was a great summary.
Sure was.
That was really, really good. Thank you, Michael. Well, over to you, Ed. Do you have a question for the doctor?
I'm sweating on this question. So I think over the course of now 31 episodes of the podcast, I've kind of started to get some very basic understanding of what neuroplasticity is. And so I think I'm all on the whole brain train kind of deal.
We'll get you a T-shirt.
but I'm definitely unaware of what the spinal cord, I guess, contributes to all of that. And so this kind of research is obviously very interesting from a cerebral palsy perspective. And I guess I look at it from the layman's perspective because it's the only perspective I can take on these things. And this study was done in adults, wasn't it?
Yes, it was. Yep.
Okay. So I guess my question about this research is it's it's something that's affected the spinal cord, but it's shrunk. Is that my understanding? Like there's a portion of it that has become smaller?
Yep, that's correct.
So I'm trying to figure out, and you probably can tell me immediately, or perhaps it's like an incredible mystery, but is it smaller because of cerebral palsy, or was it always small because they were a child and it's been measured obviously when they've become an adult, or is it because of the outcomes of cerebral palsy, the effects of them not being able to use their limbs, or the exercise or things that they've been doing? Is that a reason why it's smaller, or am I completely on the wrong track? Because I'm now physically sweating. I can feel myself sweating.
No, absolutely. I think that there may be multiple things that are contributing to that size difference. And I think that the first thing could be just downstream effects within the nervous system as a result of the insult that happened during brain development. And so kind of direct changes that are occurring within the nervous system. But as you said, a result of that is going to be that these individuals are going to be a little more sedentary. And so this lack of activity and sensory and motor experiences is going to also contribute to neuroplastic changes that can also ultimately result in these changes that we are seeing at the spinal cord. So that's a great question.
Okay. All right. I feel less silly.
Yeah.
I'm going to have a quick follow-up because I've been getting in a second question these days. Can you do something about that? Like is doing more exercise or working with physios and OTs and the rest of it, like would that change things?
I think that is a great area to investigate, because what I'm definitely particularly interested in is if these changes that we see in the spinal cord, if they are able to be changed following some sort of well-known therapeutic intervention that works. And so are these differences in the spinal cord not only able to be changed, but also are they related to some of the functional improvements that are seen following an intervention?
I feel smarter already.
Yeah, here you are
go.
asking the big questions for the next PhD, hey? That was actually really good questions. And I think you answered them really nicely there, Michael. I think that's something that we can all take away from it. So thank you so very, very much. And for everyone who's listening, who want access to the paper and who wants to look at some of the show notes for the podcast as well, remember you can head to our website, researchworks.net. There's also a CPD form there that you can fill out so you can keep a record of your PD, particularly as we're all about to get audited as health professionals. So we can help you keep a record of all of that as well.
Yeah. Michael, thank you so much for joining us from the other side of the world.
Absolutely. Thank you for having me on.
Oh, we have loved the conversation.
Yeah.
We love knowing you a little bit more, and, just keep an eye on those keys, put a little GPS on it or something like that. You can find it.
Yes.
I'll have to figure something out with that.
Well, to all of our listeners, thank you so much for listening, and we'll talk to you guys again next time. Bye.
Bye.